Well, I'm really delighted to introduce tonight's speaker, Dr. Chandra Jack of Clark University in Worcester. Dr. Jack earned her BS in biology from Rice University in Houston, Texas. She then went on to earn both her Master of Science and her PhD at Rice in the Department of Ecology and Evolutionary Biology. Her graduate work focused on microbial influences on plant functional traits. Since completing her graduate work, she's held several appointments. First, as a postdoctoral researcher in the Department of Bioengineering at Rice, and then as a Beacon Distinguished Postdoctoral Fellow in the Department of Biology at Michigan State University. She's also held an appointment as a research associate in the Department of Plant Pathology at Washington State University. Dr. Jack joined the faculty of the Department of Biology at Clark University, where she's an assistant professor. Her research explores, again, plant and microbe interactions and how those interactions mediate plant traits and fitness. And her lecture tonight is titled Microbial Influences on Plant Functional Traits in Natural and Agricultural Systems. Dr. Jack, a very warm welcome to you, and thank you for lecturing tonight.
Thank you. So there was a slight change to my talk, I think, between when I first agreed to give it. As I've just added more and more data and made modifications, I've sort of updated the overall idea, but the same idea is presented here, just modifying the title a bit. So what I'm going to be talking about today is the functional rhizosphere and how microbes and soil health influence plant fitness and the different traits that go into what makes a plant a plant. This picture, you'll actually see, I think, later on as well. It's one of my field sites on the Palouse area back in Washington State. But what I really want to talk about is how we think about the framework for understanding the relationship between plant genotype and fitness. So the conventional framework is that you think about the plant's genotype, and that that determines the phenotype, which impacts fitness and then representation in the next generation. And through this framework, microbes are often ignored or they're considered just a part of the environment. However, we can more usefully conceptualize them as what we refer to as an extended host. And by this, what I mean is when we think about plants, they are these entangled hosts. They contain a wide array of beneficial and, and antagonistic microbes that interact with each other and the plant in complex ways. The surface of the microbe, both below and above ground, hosts these microbes. Today, I'm focusing only on beneficial microbes that occur in the rhizosphere. The rhizosphere is this thin plane where the soil and root surface interact, and it's a metabolic hotspot for microbes. And what we see in this in the rhizosphere is that they are shaped by a combination of local environmental conditions as well as plant genotypic effects. And so what we see is that when you move towards away from bulk soil and towards the root itself, we find that microbial diversity decreases, but microbial density will often have an exponential increase. And this is because plants are recruiting specific microbes through the deposition of distinct carbohydrates into the soil. And then these microbes, through their host associations, play a pivotal role in the health, productivity, and fitness of plants. So knowing that there's this, that we can consider microbes to be this sort of reservoir of genes for the plant to use, it's more probably a better way to think about the interaction of the plant phenotype is sort of an expression of joint genomes, where you have an individual's fitness determined not only by its own genome, but also by the influence of its partner's genome on those traits. And in fact, you can end up getting these sort of complex fitness feedbacks between the genomes of the two interacting species. So a lot of times when we think about the extended phenotypes and mutualisms in evolution, we tend to apply that to mutualisms. So we're especially the ones that I work with, rhizobia and the legumes. So in this case, you have the legume rhizobia symbiosis, where the plant will secrete these flavonoids that are host specific, and then compatible rhizobia are produce these what we call nod factors that initiate the formation of root nodules, leading to the symbiosis that occurs between them. However, microbes do not only mediate traits that influence just the association itself, but also beyond the interaction. So these traits can include morphological, physiological, and phenological traits. Flowering time, for example, is especially found to be under some influence of microbes in some cases. So when we also think about like how microbes themselves can impact plant growth or fitness, it can occur in a number of ways, many of them often agronomically important. So for example, microbes will reduce oxidative stress through the presence of antioxidants. They're involved in osmoregulation. They can produce phytohormones themselves or enhance the production of those hormones by the plant. And they can also modify the availability of different nutrients that are required by the plant, for example, nitrogen, phosphorus, or iron. And so when we think about the modifications and how these traits can influence plants, it's really sort of interesting to know how adaptive are these traits. And in other words, can the variation of these traits influence the fitness of the plant? And so what we can do is we can use univariate regression approaches that were described by Landon Arnold to study phenotypic selection. So one of the ways is where you have your relative fitness. A lot of times it's, for example, seed production, but it can be something like the plant biomass that's produced. And you regress that against whatever trait you're interested in. And then the function of the slope is equal to the strength of selection, which determines whether or not there is an adaptive variation between the trait value and its influence on the overall fitness of the plant. So what I'm showing, and then because I'm also especially interested in whether or not there's beyond just regular selection occurring, we're also interested in how microbes influence fitness. What you can do is sort of is an analysis of covariance, where you incorporate the treatment. So in this case, the presence of rhizobia or the absence. And by looking for a significant interaction term, you are able to determine how much of the trait variation and its influence in fitness is dependent on the rhizobia or microbes themselves. And so in the top figure, I show what we refer to as linear selection or directional selection, which changes the trait value itself. But you can also model nonlinear selection by adding a quadratic term, and that changes the variance around the trait. So now we're looking at stabilizing or disruptive selection. And we find often that mutualisms are predicted to undergo stabilizing selection. And then I have this figure that you'll see quite a few times, where I'm going to discuss how individual components of it are relevant to some of the research that I'm going to show, or the results that I'm going to show tonight. So one thing that's really interesting is this section here, where we're comparing the fitness of species A and the fitness of species B, and this idea of fitness feedback. So if you recall, like for mutualisms, because the definition should be that both species that are interacting benefit, you can determine whether or not traits have the same influence on them. Because if you look at, they can have different functions for those traits. And so if the interest overall of the of the microbes are aligned, you can look at the relationship between fitness of the host plant and its microbes. So for example, if there's no relationship, when you do a correlation plot between the fitness of the plant and the microbe, you would just see sort of a cloud of dots of data points. If the interests of the two are aligned, we would see a positive slope, showing that as the microbial fitness increases, so does plant fitness. If there's conflict, which should not occur for mutualism, you would see that when the microbial fitness decreases, that plant fitness increases. And then, so as I said, for mutualism itself, we would expect overall that they should be in an alignment. However, that does not mean that just because the overall fitness interests are aligned, that the individual trait values are. So moving sort of up in this figure, you'll notice that there are trait fitness functions that point from the trait of species A to the fitness of both species A and B. There is no requirement for that function to be the same for both. And that can lead to sort of a misalignment of interest, meaning that microbes might prefer a different trait value compared to a plant. So to sort of explain that, I have this figure here. So you can imagine that if you consider the x-axis to be the value of the trait and the y-axis to be just fitness, whether it's the plant or the microbe, if the two are in alignment over a particular trait, you will see peaks that overlap each other, meaning that they have both the microbe and the plant have the highest fitness when they have the same trait value. However, there can also be conflict. So for example, let's say microbes in particular, they are found in the roots, mostly for what I'm talking about. So you could imagine that if that's where they have most of their associations, they might prefer to have a larger root biomass, whereas a plant might prefer to allocate resources more towards the shoots. So if you were to imagine for that part C of that figure, that there is a conflict over how much allocation there should be to roots compared to what the plant would want versus what the microbes would want. And then there can also be varying strength. So again, that distribution value. So maybe they do have similar means, but the distribution and the fitness differs between the two. So for example, the plant has a narrower areas of what it prefers its trait values to be for the higher fitness, whereas the microbes might just, it not matter as much. As long as they have any sort of roots, they're happy. And so that's a little bit of background about some of the statistics that I'll be using for this first part of my talk. And that's going to focus mostly on the natural systems. And so this first project is focusing on selection on these microbially mediated traits in a species of clover that is called trifolium. So in this particular picture, this is the UC Davis Marine Bodega Laboratory. And it's really interesting because in this area, there are 9 native species of clover, and they all coexist and they all share different rhizobia. So there's not necessarily where rhizobia only stick to one species of trifolium. And so in my former lab, when I was a postdoc, they would go out here and they would collect seeds, different rhizobia strains, plant tissue to bring back to the lab to allow us to do all these different experiments. And so for this experiment, I will be using one of those 9 species, Trifolium barbigerum, and its symbiont, Rhizobia leguminosarum, to answer 2 questions. And those are, which plant traits are influenced by rhizobia? And then how much of the plant phenotypic variation is due to the genotype of the microbe? And actually, since I'm not really going to answer much of the second question because I just have not gotten to analyze all of that data yet. So that's a future talk. But what I did is I set up this large scale greenhouse experiment. So this is our greenhouse back at Washington State University, where we grew one accession of Trifolium barbigerum with multiple strains of its associated symbiont. And so what we had for this experiment is 77 different strains of Rhizobia. And then we had our controls of the negative treatments. They just got phosphate buffer and then a nitrogen treatment as well, just sort of to compare whether or not getting your nitrogen from an external applied environment compared to from the biological nitrogen fixation. And then what we did is we allowed this experiment to grow for about 6 months, and we harvested the plants in 2 batches. So after 3 months, we harvested half of our plants to measure below ground traits, so focusing mainly on the root architecture. And then at 6 months, we harvested the remaining plants, and we focused, we called those sort of our above ground traits, but when you're harvesting plants, you never leave any tissue behind. So we do have root data here as well. And then the focus of my talk is going to be on those plants right now from that above ground or second harvest. And so what we first saw when we did the analysis comparing the fitness versus the traits, so that's first figure I showed you, to look at selection and the strength. And we can see that most of our traits had a strong effect on fitness. So in this case, what I've just done to make it easier is I have that S, so that slope or that strength of selection showing on the x-axis. Then I have the different traits on the y-axis. And if there's a significant selection that's acting on them, the value will be away from 0. And so what we can see is that, and it can be either positive or negative selection, it doesn't matter, as long as it's not 0, it's most likely significant. And so what we can see is that many of the traits are significant. And then off to the corner, you'll see I just have a little mini slide that has shoot biomass versus seed number, because a lot of times when you are doing fitness measurements, you want sort of reproductive success to be your measure of fitness, but not all of our plants set seed. So we ended up using shoot biomass, and that little insert just shows that there was a high correlation between shoot biomass and seed number. So we were confident in using that to be a fitness proxy. And so we're really excited about this. It was really interesting, but unfortunately, as is often the case with science, you don't always get the expected results the first time, because of all of these traits that we looked at when we were looking for treatment differences, so whether or not the Rhizobia made a difference. We only found 2 traits that showed any sort of treatment difference. And you can sort of see here, we have plant height and then flower number, where the lines are not really sort of on top of each other. So most of our traits were actually not significantly different from each other when we do a comparison of the bacteria, the negative, and our nitrogen. So that was a bit disappointing. However, so one of the things that's really great about having so many strains is that you have a lot of data to work with, but sometimes you can have too much. And so I wondered whether or not we just had too many strains to see the variation that would be present. So what I did is I started doing some cluster analysis, which allows you to kind of bin your different bacteria strains together. And so here is what I did was I binned all 77 strains into 4 different clusters. And you can see that when you do a comparison of the shoot biomass versus root biomass, each X is a grouping of strains, and you can see that they kind of fall out pretty nicely and allow us to distinguish them. And so I've started doing this analysis, and it's so the early analysis, it's promising. So this is just a linear model showing the differences for shoot biomass, where you can, or when I have just the sort of traditional 3 treatments, bacteria, negative, and nitrogen, you can see that there's not a significant difference between them. And then when you look at when I cluster in those 4 groupings and include the negative and the nitrogen, you can see that I now have a significant difference in treatment. So this seems to be the way to go. And that's what I'm working on it right now. Also, so when you're doing selection analysis, you get these really interesting results, but they're oftentimes not the greatest way to give talks, because you tend to just end up with tons of tables, and no one really wants to just, attend a talk where there's nothing but tables as you try and explain. So I'm trying to limit that, but what I've done here is I've just made a figure of all of the traits that I'm interested in. And you can see from the figures here that we get a lot more resolution and separation between all of the different lines. So what this indicates is that, that there's most likely some differences in the clusters and how they influence plant fitness. And so the one table that or quasi table that I'm going to show you, and the numbers don't necessarily matter, just the highlighting does. So here is the selection, the strength of selection values, or the traits and the p-values. And what's really important is the highlighting. So these are bootstrap p-values. And so if you have yellow highlighting, it means it's significant interaction. If it's not, it's non-significant. And so the leftmost table has just like the traditional treatment structure. And you can see that, again, we only had our 2 traits that were effective interactions, suggesting that rhizobia had a strong influence on fitness. But when we cluster it, you can see that many more traits show a significant interaction on the fitness. So this is suggests that there's a lot of variation in the different strains, and that by clustering, we can start to sort of parse out some of the differences that we can see. Also, really interesting about this data is that for most of the figures, I'm just going to go back for a slide, you can see that all of the slopes tend to go in the same direction. So they're either all positive or they're all negative. However, there is one trait where we did not see that effect. So this is the root to shoot ratio. And you can see that the bacteria and the nitrogen treatments have slopes in the opposite direction from each other. So this suggests that maybe there's some type of conflict going on, because we have potentially differences in selection values here. So again, pointing to that little diagram, the trait functions perhaps being different. And so what I did was I did a simulation that predicted the slope of, or the fitness function for both the or for the microbes and the plants for this trait. And what you see is that it predicts that there should be some conflict between the trait values. So of course, a lot of times you don't always get both sides of a curve when you're doing the trait value, so you really have to rely on the predicted simulations. But if this holds, this would suggest that there is a conflict of interest in what the trait value should be for that root to shoot ratio, which is really interesting to me. So something to further explore. However, despite this potential conflict at this one trait, overall, we did see that our host rhizobia fitness interests were aligned. So again, the positive slope between plant fitness, in this case, shoot weight, and microbial fitness, which we used the total nodule weight, and that they are pretty much in alignment. So I'm still working on analyzing this data, but we're getting some really interesting results. Some of the summary from what I've shown so far So rhizobia can modify plant traits, even ones that are under strong plant genetic control. We saw that overall the fitness interest of our barbigerum and our rhizobia were aligned. There is some evidence for conflict between some of our individual traits. And then all of that trait variation that kind of masks those differences suggests that some rhizobia may be better partners for the trifolium than others. So there are 3 directions for this research that I'm currently going in. One is to continue this idea of the extended phenotype. And to do that, we would do GWAS, so genome-wide association studies. And what this is really asking is whether or not we can find evidence of variation in the genome of our different rhizobia strains that points to the variation that we see in the traits. And so that's really interesting because then we can see, if you have this modification of the genome, you'll get a trait value of, X versus X plus 1. So it'd be really interesting to continue. The second part is focusing on that variation that I talked about. And this will be looking at partner choice experiments. So this is all 77 strains that I've laid out by their mean shoot biomass. And you can see that there is a significant amount of variation that goes from some having a significant, and then the red line is the mean biomass if you have uninoculated plants, so just sort of our control. And so you would expect that if the strains are beneficial, they should all be above that red line. And for some of those, you can see that's not the case. And then apologizing ahead of time because when I did this next figure, it flipped and I didn't quite realize it. But when I spoke about the clusters, so when you cluster and show that same figure, you can see again those nice bins. So again, suggesting that there are some partners that are better than others. So why is this important? Well, when we think about the idea of partner choice, so in other words, what rhizobia strains does the plant choose to associate with? Because remember, there's this relationship where the plant is providing carbon to the microbes in exchange for fixed nitrogen. And what we often see is, because I sort of mentioned that there can be this host specificity, but the problem is that the plant does not have perfect information. So nitrogen fixation, or in other words, how good of a partner the rhizobia is, does not occur and become known until after the plant has sort of committed to forming the symbiosis. So I made an example when I was talking to some of my students. It's like, okay, you're going into lab and you have the choice of sitting next to a student that you worked with last year or a new student. Who do you choose? And that's kind of the basis for this project. I'm trying to figure out what information is out there that the plants can use, since it's not going to be just the nitrogen fixation, since that doesn't occur, to be able to determine how they should choose a partner. Is there some sort of unknown signaling mechanism? Is it related to the specificity that can occur? Because remember, there's all those different nod factors that I mentioned. Each rhizobia makes these different ones, and it's under the genetic control. So there has to be some type of signaling or some molecular mechanism that can help the plant choose the best partner. Or at least I hope there is, because that's what I'm planning on doing. So I have this project that I set up, and where I'm growing all of those trifolium, all of those 77 different strains, so they have all become sort of familiar with it. And I took seeds from those. And now what I'm going to do is, so for those offspring, is give them a choice of 2 different rhizobia. So I'm going to give them one that they're familiar with from sort of, previously being inoculated with it, and one that they're not. And then I'm going to determine which rhizobia strain that they choose to partner with, which one will they associate with more. And then I'm also going to vary how beneficial that microbe was. So we also know the effect that each one of them has on the shoot biomass, so our fitness of the plant, in order to determine whether or not there is some sort of connection between familiarity and also the benefit of the plant, of the strain. So you can imagine that if you give a plant a choice of a strain that it's really familiar with, that has a really good partner, it should choose that one. But what if you give a plant or offer a plant a strain that it's familiar with that was not a good partner? Should the plant, sort of bank on what it has, or should it try for the unknown in the hopes that it's a better option? And then does it matter how varied that difference is? So if you can imagine on a scale of 1 to 5, so if you give the plant a choice of a 1 versus a 5, will it make a different choice if you give it like a 2 versus a 3? And so that's this experiment that I will hopefully be starting later this semester. I've started working on that as well. And then the third direction for my research, which also leads into the next part of my talk, is thinking about how we can move from this sort of theoretical world into more of an applied system. So for this, we're thinking about microbiome assembly. So what I hope that the connection that I made is that we know that individual strains or individual microbes can influence plant traits. So then what can you do with that knowledge? And for that, it's thinking about synthetic microbes or microbiomes. So can you make sort of a designer plant by only changing the microbes they associate with? And so from that, it's thinking about the abundance of particular species, how they can affect plant traits, and then how they can affect plant fitness. So in this example I have here, this was a experiment that was done by one of my friends and colleagues, Renee, who looked at the microbiome of switchgrass, which is a Department of Energy crop they're really interested in trying to make biofuels. And what you can see is that as the abundance of this one particular strain, Micromonospora, decreases, root length of a plant will increase. So that's really interesting. So by just changing the amount of the strain present, you can vary the root length of the plant. But if you look at the next figure, what you see is that root length is positively correlated with the fitness of the plant. So you can imagine that if you have a plant that has a lot of Micromonospora, it's going to have lower fitness compared to a plant that does not have that much. So if you're trying to design, especially for agricultural purposes, to increase crop productivity, and you wanted to increase the fitness of your switchgrass, you can imagine that if there are a lot of Micromonospora that are present in the soil, it's going to have lower overall fitness and productivity compared to when you're not. So if you're making a sort of a synthetic microbe system to inoculate your plants with, you would want to make sure that you do not have Micromonospora present. And so this leads to this idea of now agriculture, what we can do with this information. So this is a paper way back in 1927 that discusses sort of the ecology of agriculture in prairie sites. And the focus is on climate, topography, and soil as sort of the things that are really important to make soils deep and fertile, and sort of what is really great about prairies. So here's also one of my field sites. This is a prairie. It's in the region that we call the Palouse, and it's defined by the Palouse River watershed. It's a region in the inland Pacific Northwest and encompasses northern Idaho and eastern Washington. So all of prairie, all prairie ecosystems are endangered. However, the Palouse Prairie is one of the most endangered ecosystems in the continental United States because there's less than 1% of its original habitat remaining. Because all of those conditions and traits that make the prairie one of the most sort of biodiverse and productive environments also helped lead to its downfall, because that made it was good for agriculture. And so a lot of times what you'll see is something like this, where you might have a small swath of prairie, but it's surrounded by agriculture, especially wheat fields, which you can see. And so by 1890, nearly all the Palouse lands had been taken up and converted to wheat farming. In fact, historically, the Palouse produces twice the national average of wheat. And one of the problems is that agricultural intensification has replaced the biodiversity of the prairie microbial system, both, I guess, and also the plant diversity as well, because of the monoculture, which has led to sort of drastic physiochemical and biological changes to the soil. So what we can also see is that this period of agricultural intensification, which is this period from about 1960 on, where we had a combination of new breeds of crops and also the advent of synthetic fertilizer. And you can see that over this time period, from when the Haber-Bosch process, which produces synthetic nitrogen, was found, you see this marked increase in application of fertilizers. In fact, they went up over 800%. And this, on its face, seemed like, it was the right thing to do. It doubled our global cereal production. Without this applications, we would require over 4 times as much land to meet our current global demands. Unfortunately, it's of an extremely energy intensive process, and it's also environmentally unfriendly. So it leads to increased aquatic pollution, and also greenhouse gas emissions have changed as well. So it's very problematic, this process. And now what we see is sort of this growing cry for how to make agriculture more sustainable, or sort of an in-depth look at what we're doing for agriculture and whether or not we can sort of change what we're seeing. And so now I'm going to sort of show what some of the what has been some of the consequences of agriculture and our soil and its health. This picture here shows an area of the Palouse. It's a USDA owned research station, Cook Agronomy Farm, where they have been monitoring soil health and wheat production for over 20 years. So as little as 70 years ago, the Palouse soils were near neutral. However, there have been these continuous applications of synthetic nitrogen, and you can see that the soil of the Palouse is now extremely acidic. So it's below 4 in some locations. And I'll explain the different parts of this figure, but really, one side is where they have continued to have traditional farming methods, and on the other, they've started to move away towards a more what they're calling aspirational model. And so we're going to see what that entails. So one of the processes that really influences the soil and the soil health is the soil structure. So a lot of times when you have wheat farming, you will have what we refer to like the tillage, where they're constantly breaking up the soil. And so conventional tillage, you can see that it tends to really crumble the soil compared to even reduced tillage, where you have much larger chunks. And when you have those larger chunks, one of the benefits is that it helps keep some of those microbial networks intact. And that, plus the combination of different plants that are found, means that you have increased biodiversity. So for example, if you do a comparison of prairie systems and agricultural systems and look at the microbial diversity and abundance, you'll see that in agricultural systems, fewer microbes and much less diverse. In addition to that, what you'll also see is that agriculture has changed the soil organic matter of the field. So in other words, just like how much organic matter, which is sort of a proxy for the health of the soil. So again, we have our aspirational side, and then BAU is just the business as usual, meaning that traditional farming methods that they have compared to the reduced tillage on the east side. And you can see that by reducing the tillage, you increase the soil organic matter, leading to a healthier soil. So just as an idea of what soil organic matter does, and then tillage and how that can influence the overall structure of the plant, I have a figure here. So an agricultural field, which if you just place a clump of soil in some water, you can see that it quickly disintegrates. Whereas the prairie soil, that same lump is in there, but the water is relatively clear. The clump is being held together by the organic matter, the microbes, humus, just everything to help keep that soil intact, meaning it's overall better structure. It's able to hold on to nutrients, and just is a healthier, more fertile soil. So altogether, when you have the use of tillage, synthetic fertilizer application, monocropping, what you end up with is this nitrogen pollution, where you have all of the nitrogen from a farm is dumped on the fields. The plants are not able to take up everything. It runs off, enters the groundwater, becomes contaminated, leads to also air pollution as well. So it's a problem. However, we have another problem going on as well. The year 2050, that is our global crossroads for food production. So the year 2050 is when we are expecting there to be about 10 billion people on Earth. And this is when we would expect our global food demand to outpace our agricultural crop production. We would need to increase our production by 56% in order to meet the demands. And so that's problematic because we've already shown that our synthetic fertilizers aren't that efficient. They require multiple applications, which means that it requires a lot of energy to produce them. And that leads to an increase in our environmental pollution. So we need to do something, we need something else, right? We need to look at this from a different perspective. And that has led us to what we refer to now as the green revolution, where there's a focus on sustainable intensification. And being more, just using, changing our agricultural management styles so that they are more sort of environmentally and ecologically friendly. And that's, again, what has been occurring at the Cook Agronomy Farm. And what you can see is that even though the soil is still really acidic, but the increase in the soil organic matter, and what's happened is that the use of nitrogen is much higher in our aspirational field now, and it's led to a 35% increase in the crop productivity of our wheat. So if you can get people and farmers to buy into this, over time, eventually you can have a sort of a wheat crop productivity that is better, that it's higher and then also better for the environment. And part of that is being aware of microbes and the benefit that they can have. So trying to bring everything back together. So when we're thinking again about the green revolution, we have a comparison between legumes and then cereal crops. So legumes, which I sort of focused the first part of my talk on, that's what the trifolium falls under with that symbiotic relationship. They're the second most important crop, and they input over 44 tetragrams per year of nitrogen into natural and managed systems. So this is nitrogen that is produced through biological natural processes. And then we compare that with our cereal crops. So our cereal crops are our problem, all of our grains and whatnot, because they are responsible, or they receive 60% of our synthetic nitrogen. They are unable to form those symbiotic relationships. But there are these other nitrogen fixers, so diazotropes, that are free living. So they don't form that really close association, but they are in the soil, and they do provide nitrogen into some agricultural systems. So you sort of think about, well, what can we do to enhance the soil to make it a better environment to increase their abundance, for example? And that's where we're starting of, like, how can we have a healthier soil? How can we understand and take what we know from prairie soil and the biodiversity there and apply it to agriculture and our different management systems. And so for this project, I had two questions. So one, how does prairie soil affect wheat growth and flowering time? And then whether or not it's microbes that are involved. So is it just something that's inherent to prairie soil itself, or can we parse it out into the microbes? And so this project I did my second year of sampling last year. And I have different field sites. We tried to pair them all up. So this is the Palouse area, and we did sampling of prairie and agricultural sites. So the agricultural sites were our black dots, and our prairie sites were our different gray ones. We went out, we sampled soil from all these different fields, and we tried to get ones that were really close. And so one of the things I mentioned is that prairie was really endangered in the Palouse region. So where can you find it? And one of the answers is like there are some preserved graveyards. So we got permission. We have the picture there. That is Pitt Cemetery. It's a very old graveyard that they have allowed to sort of be reclaimed. So it was a great place to sample. And then as well as some of our other sites that were just landlocked by wheat fields. So 2 years of sampling there. And then we brought everything back to the greenhouse to set up a controlled set of experiments. And we actually broke our experiments down into 3 different ones. I'm only going to really talk about 2 of them today, because again, early in stages of analysis. So the first experiment, we had our soils, and we did a experiment that was 50% field collected soil and 50% sterile sand that we grew our wheat plants in. We allowed our plants to grow for about 8 weeks. So one of the really funny things about this is that I got USDA funding to do this project, and a few months later, so I was planning to do this whole thing when I was out at Washington State. And then I found out that I got the job at Clark. And so that kind of changed everything. So we set up this experiment. It was also sort of interesting because we were waiting on the USDA to give us permission to sample at Cook Farm, and they didn't get back to us. And then one day they were like, okay, if you want to go out there and sample, you have to do it tomorrow because we're getting ready to spray for an invasive weed. So we had to do that. And so because you also want all of your soil to be around the same age as possible, that means we had to collect all of our sites. So it ended up being 55 subsites, set up this experiment, and then we harvested June 30th. And then July 8th, I left to drive across country to go to Clark. So left everything pretty much behind. It was like having to have things shipped. It was a really great experience. It was completely hectic, but some of the analysis that we did find was pretty interesting. So when we had this 50-50 mix of microbes or prairie soil and sterile sand, what we saw was some interesting results and some unexpected. So we found that prairie soil does not enhance wheat height. It does increase the shoot mass of the wheat, but we did see that prairie soil accelerates flowering time of our wheat, which can be a really positive thing when you consider how unpredictable the Palouse is in terms of weather. The first year that we sampled, it was a drought. The second year, they had historic rain. So you can imagine that if you can flower earlier and get through your productivity or your life cycle to increase your overall productivity. So some mixed results, but pretty interesting. So there is some positive influence of prairie soil. And the next question was, well, how much of that is due to the microbes? So because we had such a high percentage of prairie soil, you are unable to sort of parse out microbe from soil conditions. So what we did is we decreased our ratio. This time we set up it was at the same time, but this experiment, 1% field soil this time and 99% sand. And then again, with our, we grew our wheat. And what we found is that the prairie microbes, when they were present, did not enhance the wheat height. So there was very little difference. And we found that the prairie microbes did not enhance wheat shoot mass, so no change in productivity, which is what we had especially predicted. So we're not really, that one hurt. But we did see that there was an increase or an acceleration of wheat flowering. So microbes are making the wheat flower sooner, which, still is a benefit. So what can we conclude from this? We know that prairie soil promotes wheat growth, but two possible conclusions. Microbes are not involved, which seems to go against what we would predict and what previous research has shown, or we had some methodological issues. And what we're leaning towards is option number 2. I have no problem admitting that we need to change our methodology. It's part of science. So what we found, there's this paper that came out where they looked at the biodiversity or the diversity of soil, so whole soil versus different fractions of soil. So if you were to compare that to 5% soil, 1% soil, 0.1, and just like a wash of soil. And what we could see here is that when you have 1% soil that you use, it is significantly different in terms of the diversity of microbes that are present when you compare that to the sort of whole soil. So what this suggests to us is that we need to redo this experiment and up to 5% soil transfer. Because again, we don't want to use 100% because you still need to be able to separate prairie soil out or the soil conditions from the microbes. So if we go up to 5%, we're hoping that we'll see some of those microbial effects that we predict. But remember that the whole point of this project is not only to see the effect of microbes that are present in the prairie, but also think about how we can improve natural or biological nitrogen fixation. And so what we did at the same time is we set up an experiment where we inoculated our plants with a diazotroph or a known microbe that is able to fix nitrogen. And we used our 1% field soil with our 99% sand. And we have some promising initial results here. So on one side, we have the inoculation with our Diazotroph Azotobacter benelandii. And on the other side, it was uninoculated soil and allowed our wheat to grow. And the different colors refer to, we just gave the plants synthetic nitrogen. And what we can see is that the presence of the diazotroph increased the dry mass or the crop productivity or fitness of our wheat, regardless of whether or not we had a synthetic nitrogen fertilizer or if we just had prairie soil. So what this suggests to us is that if we are able to make these soils, enhance them to make them friendlier for microbes by changing some of our management systems, decrease our tillage, and use less synthetic fertilizers, any sort of diazotrophs or nitrogen fixers that are present should have an overall positive effect on plant growth. And that's where we're moving towards this for this particular project. So where are we going with this one? Well, we have all of that soil. We have now, so we have 55 sites from 2021 and another 20 from 2022. And we are going to isolate the microbes from all of these soils and then test them on our wheat plants to look for diazotrophs that not only enhance plant growth, but also are good nitrogen fixers so that we can move towards making sort of those synthetic microbiomes that I talked about to enhance diversity and increase crop productivity. And so altogether, what I really want my take-home message to be is to think about how plant traits experience strong selection from microbes, that they can be impacted by trait variation differentially. So some microbes and fitness are not necessarily aligned in all cases, but we know that we can have selection on plants that can change microbial abundance, which can then feed back on the fitness of the plant itself. This will allow us to design synthetic microbiomes that we can use to increase our plant fitness. And if we can increase plant fitness synthetically, we can apply that knowledge to moving towards wholesale changes in agriculture and moving towards a more sustainable way of feeding our growing population. And so with that, the acknowledgements, my friend and collaborator, Renee, my current lab, some of my undergrads who I took with me back to Washington last summer. It was quite an adventure. We actually got stuck in a field and had to get towed. And then members of the Friesen lab who were instrumental also on this process. And with that, I guess, thank you.
Thank you, Chandra. That was absolutely fascinating. I learned so much. And we are already beginning to have questions in the chat. If I might start with one, I had jotted one down rather early in your talk. I'm so interested in all the different traits that can be influenced by microbes. And particularly, many of us, I think in the audience, have read papers about climate change influences to flowering time. I hadn't really considered microbial influences to timing of flowering in plants. And I wondered if you know or could explain a little bit about the mechanism through which microbes are influencing flowering time. Is it something to do with hormones in the plant, or?
That's what we think, that it's sort of changing some of those phytohormones that are responsible for growth in combination with making it a better, so microbes, they'll increase the phytohormones to help growth. They also will bolster the nutrients that are available. So perhaps having more of those microbes earlier on means they can get to flowering earlier. And then also because it can also attract some of the pollinators as well by making modifications. So all things that can help with the flowering time.
Thanks, Chandra. Okay, let me begin to read through some of these questions for you in the chat. Okay, so first we have a newbie question that says, relating to what you had mentioned, the partnering. How do plants pick and choose the microbes that hang out in the immediate vicinity of the roots? Sounds like you're going to be looking into that soon.
Yes, that is really the question that everyone wants to know, because again, it's the forming of the symbiosis. It's especially important for when you're forming a symbiosis, because the microbes will adhere to the root, and then tissue is actually going to form over it to form that nodule to allow the exchange of nutrients. And so you can imagine that's a pretty energetically costly process. And so we don't really know what specifically it is that will allow the plant to determine who enters the nodule or who they'll form a nodule with. Like, we know that there is some competition between strains themselves, which influences it. And we think that there are also signaling that it's unknown right now between the microbe and the plant, but we have no idea what that signaling is. Because again, any sort of signaling that's related to nitrogen fixation does not occur until after the nodulation process.
I see. Well, thank you. All right, here's a question from Matt. Is there much variability in the global distribution of Rhizobia species?
There is. So for example, just in that one area of a bodega that we were at, we were able to get just those 77 different strains, and that was just doing a smaller transect. So you can imagine that when you extrapolate that across the country, that it would, the scale of biodiversity is probably pretty big. But one of the problems that we're also finding is that people tend to take microbes for granted. So oftentimes, unless it's a specific experiment, no one really knows what microbes are present, which is another problem if we're trying, like you often see now advertisements for, wonder inoculations. We can add this. And the problem is that you don't know what's already there in the soil. So if you're adding something to it, then there can be all sorts of ecological unexpected results that may occur. And So it's more research that needs to be done is really identifying who and what they're doing.
That's so interesting. And Matt had a follow-up question about ecological results too. Um, is it known whether the presence or absence of certain rhizobia species can influence a non-native plant species' ability to act in an invasive manner?
Yes. So actually, there's a related species, Medicago polymorph. It's a sister species of Trifolium. And one of the predictions is that its more general ability to form these associations with rhizobia is one of the reasons why it has now spread and become a cosmopolitan species, because it was originally around the Mediterranean. Now it's found everywhere. And so we often think that it's because invasive plants have less specificity with the microbes, and is one of the reasons why they're so successful.
That's so interesting. Okay, here's a question from Lisa. Are fungal partners, mycorrhizae, for example, important in the prairie ecosystem?
Yes, they are. We are considering in our next set of experiments also looking to see what fungal partners are there. The issue is trying to separate the fungi and add them directly. It's a bit harder, but we do know that fungi do play an important role as well. So when we do our whole genome sequencing, we're probably going to look for both microbes and fungi.
Great, thank you. All right, Neela has a question. How can we use this information you presented about microbes for reestablishing rare plants?
So that's an area that we also now see. I guess one of the first things is to let what grows up. So, and then increase the diversity of what's already present. Try not to add soils necessarily that are not local to your area. If there are related species that maybe, or environments that are more similar to what the rare plant is used to growing in, starting there, maybe sort of working your way out. But I think it's going to be difficult if you were to just automatically plant a rare species into a field, for example, just because of the potential competition, both at the plant level and then whether or not those microbes that associate with it will be beneficial to those plants. So I think it's probably a long process that requires a lot of thought and thinking about what soil you're using and how you can move it.
Yes, that makes sense. There's a related question from Jessa here, and you might have a similar response and maybe not. So let's hear what you think. Based on your research, what role do you think inoculation should play in natural areas restoration?
I think I'm very hesitant about the use of inoculations because even within a small transect, you're going to see such variation. And again, one of the problems is that we tend to take them for granted. So because we don't know what's there, it's really hard to say what you should add or how you should modify the soil. But I think that one of the focuses should be on making sure that your soil is as healthy as possible. So by decreasing the disturbance and also trying not to utilize any sort of synthetic fertilizers, herbicides, and pesticides.
Great, thank you. All right, here's a question from Nancy. Do Palouse prairie fires impact vegetation as much as in Weaver's tall grass prairies?
So I know that fire is a pretty does have a pretty strong effect out there. We've had the past few years some really bad fires, some bad conditions. What it specifically does to the microbial surface, I'm not entirely sure. That'd be really interesting to look into to see how fire influences the microbes. I sort of imagine that if it's burning off the top plant layer, whether or not that then leads to more organic matter that could decompose. So that could increase microbes, but that would just be me guessing.
Great. I'm going to take one from Dan here. Can you speculate about how microbial communities affect competitive interactions among plants and how these communities may be changing in response to climate change, for example?
Oh, that's a good question. So we do know for sure that there is a direct impact of microbes and their associations with plants and increasing competition between plants. Because again, that's one of the ways that we know that invasive plants, for example, have managed to become established in environments. So if you could imagine that you have a set of microbes that associate with one genotype, and they not only have a higher impact on the fitness of those plants, so they can outcompete other plants, but it's possible that the microbes themselves could outcompete the microbes of those other plants as well. So it's competition on many different layers.
Yeah.
So both above ground competition influenced by the microbes and the microbe interactions that will also change the outcome of the plants.
So complex.
Yes.
That's really interesting. All right, here's a question from Bob, commenting on Lisa's question about the mycorrhizae. There are papers on mycorrhizal transfer of phosphorus between big blue stem grasses at Kansa Prairie in Kansas using stable isotopes. Are some or many of the Rhizobia types archaebacteria rather than eubacteria? If so, what does that tell us about these critters?
So I believe most of our Rhizobia are eubacteria, but don't hold me to that. And we do know that there is some diversity in the species that are able to fix nitrogen. We are finding more because for a while we thought it was a very narrow set of species that could fix nitrogen. And as we've done more sequencing and found, so there are genes that are involved with nitrogen fixation, NIF genes. And as we've done sequencing, we've found more strains and species that have those genes. And so therefore likely to do nitrogen fixation, just need to be tested. So it's an expanding area, different species. So it's really quite interesting because not only are we now learning more about different species that can fix, but we also are learning that they can fix in different ways because they might have different nitrogen fixing complexes that are involved. So there's, yeah, the whole biochemical lesson that I could go into, but yeah, there are different pathways.
Well, thank you for taking so many questions from our super engaged audience here tonight. I can tell that everyone is really fascinated by your research. Would you be willing to take one more, Chandra?
Definitely.
Okay, Lisa had another one. Most native prairie species are perennials, but the crop plants you've worked on are annuals. Do you expect that perennials and annuals might have different mechanisms of microbial interaction?
I think that it's possible for some traits, but I think for other traits, like for example, enhancing vital hormones or even just acting as competitive, allowing for competitive exclusion of sort of pathogens, in that sense, they can behave similarly. And so we are able to at least generalize some of the results that we see.
And here's just a little quick one that just came in. Do you anticipate doing any research on local New England agricultural systems?
I always get that question. And I can honestly say not for the next 2 to 3 years, because I got the grant for the Palouse area 2 years ago now. And then I got the grant for the partner choice in May. So since I'm a new lab also, I'm just still establishing myself. I don't have grad students yet. So maybe in the future, when I get some of those projects off my lap, it would be interesting to explore, since it's a completely different agricultural system out here. I've been out to Harvard Forest, so I'm excited about that possibility, but I just know that not anytime soon.
That seems entirely understandable. Well, thank you again for answering so many questions from our big audience tonight.